Hello, my dear students. So this is the revision chart 5, and we'll be talking about an important topic. And this has been asked many times in exam. So you have pollen grain, structure, function, and viability. Pollen grain is considered as male gametophyte. The yellow powder you see in the anthers is pollen grain. High morphological diversity is shown by the pollen grains. That structural diversity is there. They have different structures. Then the size is given in your textbook, 25 to 50 micrometers is the size of pollen grain. Then it is usually spherical in size. There are two layers, exine and intine. Exine is made up of one of the hardest substance, that is resistant substance, that is your sporopollenin. Sporopollenin, it is not digestible by any of the enzymes which have been discovered so far. So the composition of the sporopollenin is not known. Some say that it is made up of phospholipids and phenols and other things. But why is the composition not known? Because it cannot be digested. It cannot be the components of this sporopollenin cannot be extracted properly, because any enzyme, alkali or acid, cannot digest this sporopollenin. That's why its composition is not properly known. Then intine is made up of cellulose and pectin. It's an internal layer. Then you have pollen grain. On the pollen grain outer side, you have germ pores. Germ pores are the actually locations where you do not have this sporopollenin deposition. So through the germ pore, pollen tubes will emerge out when the fertilization has to happen. When the pollen grain is deposited on the stigma, pollen tube will be formed. So those pollen tubes will be emerging out through these germ pores. But the pollen tube is made up of intine. It is an extension of intine layer. So, and because of this sporopollenin, you can preserve the fossil preservation can be done, and scientists have discovered the fossils of many years, and those fossils were viable also. So cells in the pollen grain, you have one big vegetative cell and small generative cell. You know that pollen grains, they can shed, they can days at 2-cell stage and 3-cell stage. Now, what is the science behind it? Why some pollens are released at 2-cell stage, and why some pollens are released at this 3-cell stage? Your textbook is writing 60% of the pollens they days at three cell stage, and then 40% they days at 2-cell stage. So this is actually evolutionary adaptation. This is an example of evolutionary adaptation. In this case, 60% of the pollens they have sorry, 2-cell stage is this and 3-cell stage is this. They days at 2-cell stage, and they delay division. Delaying division reduces metabolic load during pollen transport. And the pollen grain only contains one generative cell and one vegetative cell. So it is smaller, lighter, and more viable. For longer distances, it can be dispersed. Then when division occurs, generative cell divides after pollen lands on stigma and grows inside the pollen tube itself. When we talk about 3-cell stage, that is your 40% of the pollen grains. Here, why these pollen grains days at 3-cell stage? There is a reason, because of the fast. Because you see, if you already have two these nuclei of this two male gametes, then fast fertilization can happen. Two male gametes are already ready to fertilize again polar nuclei immediately. So common in plants where this is common in plants where pollination is frequent and rapid, or where pollen does not travel far, like this pollination by insects, this pollination. So this is very important thing that you must remember. Now, pollen viability means for what time the pollen remains active. It can grow. And for example, in your textbook, for rice and wheat, it is 30 minutes. Some plants, they remain viable, pollen remain viable for months. You can store the pollen grains. You can do cryopreservation. Cryopreservation can be done at minus 196 degrees. This has been asked many times in MCQs. Now, what is here? This is microsporogenesis happening. I told you that there is pollen mother cell, PMC. This PMC from the sporogenous tissue itself. This PMC undergoes meiosis 1. It forms 2 cells. Then it undergoes meiosis 2. Then it forms 4 cells. So this is your pollen tetrad. This is after meiotic division second. This thing has been formed. Now, after this is given in your textbook also. So after this, you take one pollen. This is one pollen which we have taken in the textbook. So this is outer layer, this is inner layer, and this is inside. Inside there is one cell. So then these are the vacuoles which are present, and this is the nucleus of the cell. Then a symmetric spindle formation happens over here. What happens? There are two cells which are formed. One is vegetative cell, another is generative cell. Generative cell is small. It's a upcoming male gamete, and a vegetative cell is the nutritive. It's nutritive in nature. It will provide the nutrition. So this is what happens in pollen grain. What is structure of pollen grain? What are the functions, and what is viability, pollen viability, and what is microsporogenesis? This has been explained in this diagram perfectly. So next video I will bring maybe today itself, and we'll try to finish this chapter within 2-3 days. Thank you.